Assalamualaikum. Today our group presenting a comparative review of two recent scientific studies that main focus on genotoxicity and water quality for the environmental health. As we all know, water is very essential for all living organisms, including us. However, the presence of contaminants like both logical and chemical toxins poses ongoing risks that require vigilant monitoring and assessment, which is very crucial to recognize these threats in order to safeguarding our ecosystem management and public health protection. So, in these two scientific articles, we will review the comparison but interconnected approaches to the water quality analysis. We will also examine the methods, findings and implications of both articles and highlight how each studies can contribute to our understanding of water issues and the importance of proactive testing monitoring genotoxicity in protecting public health and ecological system. By the end of the presentation, we will gain insights into how different pollutants can affect the water sources and how diverse testing methods can help to evaluate the potential risks. First and foremost, let's undergo the main objectives of both articles. The aim of the first article is to identify the type of cytogenetic and genotoxic effects caused by cyanobacteria toxins that are blooming in aquatic ecosystem and to determine whether cytogenetic effects depend on the complexity of the organization of ecosystem and the composition of their biotic components. These toxins can cause genetic damage in aquatic organisms and potentially impact humans too through the contaminated water sources. This study used bioassays and microcosm experiments to simulate natural conditions and assess the genotoxic effects of these toxins. While the second article is to evaluate groundwater toxicity from 7 different wells in Yucatan, Mexico, and to examine its relationship with some contaminants, which is pesticide, nutrients, and pH. This study used the allium cepa bioassay to analyze chemical contaminants like pesticides and hydrocarbons. In this part, by comparing these objectives, we hope to see how different scientific approaches come together to give us a clearer picture of biological and environmental issues.
The first article focused on cyanobacteria genotoxicity studies. Cyanobacteria bloom are increasingly common due to the climate change and eutrophication. These bloom produce toxins such as microcytins, which are not only harmful to health but also can damage DNA even at a very low concentration. While we know these blooms are toxic, we still have a very limited information on how they will affect organisms at the DNA level, especially in a natural environment with different aquatic species. This study has aimed to assess genotoxicity using the allium cepa test in 3 different microsome setups. It also explored how the presence of different organisms influence the cyanobacteria abundance and the extent of the DNA damage. This study has designed 3 experimental microscopes: zooplankton only, zooplankton with amphiphod, and zooplankton with fish. Each of these setups was exposed to a natural bacterial bloom, which mainly consists of microcytes and dolicosperma. The study has measured cyanobacteria abundance and biomass for each of these setups, microcytins concentrations, and genotoxicity using the allium test, focusing on mitotic abnormalities such as polyploid and C. mitosis. Key findings show that genotoxic effects occur even at microcytins level below the WHO safety limits of 1 microgram per liter. Study also shows that fish microsome has the highest sustained cyanobacteria and DNA damage. In contrast, amphiphod were the most effective in reducing both cyanobacterial presence and genotoxic markers. To conclude this study, cyanobacteria bloom can cause anogenic effect like spider damage, even at a toxic concentration which are considered safe. These studies show that ecosystem composition has a major impact on bloom dynamic and genotoxicity. Fish tend to support a higher cyanobacteria level while amphipods show to suppress them. The implication of this study are very clear, which is genotoxicity should be a key factor in water quality monitoring in every water bodies. Ecological management strategy should take into account the biotic structure of aquatic system. Last but not least, the allium test proved to be reliable and cost-effective tools for early detection of genotoxicity in water bodies.
For Article 2, this second study focused on evaluating the potential toxic effects of various contaminants present in the groundwater of the Yucatan, Mexico. Groundwater samples were collected from 7 monitoring wells during September and October 2021. These samples were analyzed for chemical pollutants such as pesticides and hydrocarbons. To assess toxicity, researchers employed the Allium-Cepa bioassay in which onion bulbs were exposed to the groundwater samples. For the chemical analysis, the data presented in the article show that wells located closer to the coast exhibit higher nutrient levels, indicating a risk of eutrophication. The highest concentration of pesticides detected were delta HCH and chlorpyrifos. A notable correlation between DDT and caffeine was also observed, suggesting contamination from urban and agricultural runoff. Among the analysis PAH, BK fluorantine and BB fluorantine were the most dominant. In the Allium-Cepa bioassay, no nuclear abnormalities were observed in the rock cell. Furthermore, there were no significant difference in the mitotic index, rod lengths, oxidative stress, or biomarkers of B entrance inhibition between the test sample and the control, indicating no observable toxic effects. The results show low levels of pollutants, all within safety limits, and no observable genotoxic effects or cellular abnormalities in the plant. Overall, the findings suggest that the groundwater is currently safe, although continuous monitoring is recommended. Despite the presence of certain contaminants, the findings indicate that groundwater in the sample areas did not show any toxic effect detectable by this bioassay. This method proved valuable for ongoing environmental monitoring and assessing water safety in regions with similar ecological and geological conditions. A few among the strengths is the study successfully combined chemical analysis with biological assessment, giving a more holistic view of water quality. Also, the Allium-Cepa bioassay proved to be low-cost, sensitive method ideal for regular monitoring in developing region. However, there are some limitations as the sampling period was relatively short, only covering September and October, which might miss seasonal variation in water quality. Additionally, the bioassay only used one test species, which may not capture all potential ecological effects. Finally, while no visible toxicity was observed, the method may not detect sublethal or lethal effects.
Now let's take a closer look at how these two studies compare and contrast, highlighting both their similarities and differences. First, their main focus differs significantly. The first article centers on the biological impact of microcystin, which is the toxin produced during a harmful algal blooms, with an emphasis of their genotoxic effects, which is their ability to damage genetic material in aquatic organisms. This research aims to understand the ecological and potential health risks associated with natural toxins in water body. In contrast, the second article investigates chemical contaminants in groundwater such as pesticides, hydrocarbons, and nutrients by using these bioassays to evaluate the chemical safety of regional water sources. Its focus is on detecting pollutants from human activity and their potential health implications for local communities. They also have their own objective, which is the first article aims to understand the ecological and potential health risks, that are associated with natural toxins in water, while the second investigate chemical contaminants in groundwater such as pesticide and hydrocarbon by using BIO6. It is to evaluate the chemical safety of water sources. From this, we can see that both articles actually has similar general aims to evaluate potential risks that are associated with water pollutants. Next, when it comes to the methods, both studies utilize BIO6 techniques and also use the same indicator BIO6 species, which is allium separatives. To be specific, the first article relies heavily on ecosystem simulation experiment and genotoxicity test, and which is the researchers try to mimic the natural condition and assess the genetic image. The second article primarily used the allium separ BIO6, which observes root cell division in onion bulbs exposed to the water sample. This helped to detect chromosomal and cellular damage caused by the chemical pollutants. On the other hand, their findings offer interesting contrast as well. The first study found that even low concentration of microcystin cause significant genetic damage in aquatic organisms. This implying a potential risk to aquatic ecosystem and possibly humans too if contaminated water enters the water sources chain. It emphasizes the danger of biological toxin accumulating in water bodies. However, the second study generally found no significant genetic or cellular damage in the water sample tested, suggesting that current groundwater sources are relatively safe from chemical pollutants at the sampling sites. Nonetheless, it highlights the need for ongoing monitoring as pollutant levels can be fluctuate over time. In both similarities, in this article, they also utilize quantitative data collection, which is both generate measurable and quantitative results, such as levels of genetic damage and chromosome aberrations that can be statistically analyzed. In this diagram, we also include the implication of each articles that highlight two important points, which is the first is the biological impacts of natural toxin like microcystin can be severe even at low levels and require vigilance to prevent the ecological and health crisis. While the second is the chemical pollutants are currently at acceptable levels in some cases, also need the consistent assessment because they can pose serious health if the concentration fluctuate.